Hello everyone, my name is Philip. I'm CTO and co-founder of Ablacon, which is now Cortex, and I'm happy to share with you about electrographic flow mapping to visualize complex atrial arrhythmias. These are my disclosures, and I would like to start with a real-world example. This is a patient from our Flow AF study. The patient had an initial PVI, but came back after 3 months in persistent AF. As per protocol, all 4 PVs were checked and confirmed to still be isolated. The EP then performed electrographic flow mapping with a 64-pole basket catheter. This is a picture of the navigation system showing the basket in the left atrium. And on the right are the recorded electrograms that are input to our software called OptiMap, formerly known under the name AblaMap. This is a screen capture of the current version of the software. The OptiMap system is 510(k) cleared and actively used at 15 clinical centers in the US and in the EU as part of the RESOLVE study. When you make a 60-second basket recording, the software breaks it down into 2-second segments. For each segment, we estimate a vector field that models the dominant action potential wavefront propagation. This is what we call electrographic flow, short EGF. And you can see this in the right panel. This is projected onto a 3D basket visualization. So, for example, it shows electrographic flow from electrode 1 to electrode 3. So what does this mean? Electrographic flow tracks gradients in the EGMs in space and time. As you can see, these EGMs are not easy, so this would be a challenge for traditional activation mapping. And I'll show you more details later. You can scroll through the segments like this and get an impression of what this patient's AF looked like during this minute. And you may notice this bright spot at the top. This is a location where our algorithm detected divergent flow. We call this a source of electrographic flow. As you can see, this phenomenon is not always active during the 60 seconds. The bars at the bottom show you the source prevalence per segment. The map on the left summarizes all phenomena found in the recording. The EGF source was identified in 31% of the time, resulting in a red spot in the summary map. This location corresponds to the roof of the left atrium near the LSPV. And as you can see in the timeline on the left, the source was highly reproducible in 3 successive recordings. This was the only source above threshold that we found in this patient. And according to the protocol, this source was ablated. So let's look at the EGF map that was done just a few minutes later after the ablation. As you can see, the source is gone. You can already see the altered electrographic flow at the location where the source was previously. This patient has been free from AF since this procedure for more than 12 months. To illustrate the principle of electrographic flow mapping in some more detail, I would like to show you an in silico experiment. We used a highly detailed 3D biatrial industry standard model. The model has a realistic fibrosis distribution in both atria. Arrhythmic episodes were induced by using the pacing at the end of the effective refractory period protocol, resulting in self-sustained AF. We now placed a virtual basket catheter towards the roof of the left atrium. These are the electrograms we get when we make a virtual recording with this basket catheter. We now loaded these electrograms into OptiMap to compute electrographic flow. This is the EGF map we obtained from the electrograms I just showed you. The EGF map clearly shows the breakthrough at the fossa on the septum. Using the 64-pole basket catheter, we get a global view of the electrographic flow that allows us to localize this phenomenon. The electrode separation and to a certain degree the wall distance is accounted for by the EGF algorithm. So we thought, what would we see if we place an HD grid at this position? On the right you can see the bipolar electrograms corresponding to the HD grid electrode locations. The trained eye may or may not see some signatures that look like divergence or something similar. But just for fun, we computed EGF from this HD grid recording and saw this pattern. The breakthrough source is clearly visible as well. But if you move the HD grid catheter just a bit off the source, this phenomenon becomes way harder to see than from the panoramic basket map. We believe that the strength of our approach is that we can get a panoramic map from the basket very quickly, within about a minute. On average, we identify 1 to 3 sources per patient, which are highly actionable, as I can show you in a second. The reason for this is that we developed our technology optimized for outcome. We really want to make our technology a white box. We want to make it transparent and understandable for everyone. That's why we published a white paper with the great David Haeusser. It contains all the details about our algorithm. At a high level, our data processing pipeline consists of a sequence of modules. For example, a high-pass module, a QRST compensation module, the flow estimation, the divergence detection, etc. We worked with a number of great EPs who shared with us data from a large number of patients, including information about the outcome of their procedures. We then optimized the hyperparameters of our pipeline so as to maximize the predictive capability of our source activity metric. At the end, we froze our parameters, and we determined a threshold for the EGF source activity value of 26%. What does this number mean? If you take all the retrospective procedures and look for the last recording per procedure, you can ask, do we still see a source above this threshold, yes or no? Those patients who had no such source above threshold at the end of the procedure, we put in the no sources group in this Kaplan-Meier plot. And the other ones who did have a source above threshold landed in the blue curve. We were quite encouraged that the concept of EGF sources means something. And in order to prospectively validate our method, we did the FLOW-AF study. And it's important to mention that for the FLOW-AF cohort, we really picked the toughest patient group, those that were not cured by PVI alone in an index procedure. In this subgroup, we were able to demonstrate that EGF-guided ablation results in a more than 50 percentage points higher rate of freedom from AF. This shows the value of EGF-guided ablation on top of PVI. So if we extrapolate this to all comers and imagine we do PVI plus EGF-guided ablation, just one procedure, the positive effects of PVI and EGF would add together. PVI would have a recurrence rate of about 1/3. That is our subgroup. And in this subgroup, we reduce the recurrence rate again to 1/3. So the total recurrence would be 1/3 of 1/3. Anyway, the added value of EGF is also recognized in this 2024 consensus paper, where it says, adjunctive ablation of electrographic flow sources resulted in significant improvement in 1-year freedom from AF. That's all. If you have any questions, please have a look at our website or come talk to us. Thank you very much.